Shri Sai Ram. Welcome to Shri Sathya Sai Loka Seva Gurukulam. Today we are continue to learn building blocks of life, cell and tissue. Part 6 we are going to learn today. As usual, we will start with the answer to the home assignment. Now, the first question asked was, what are the events in mitosis? In mitosis, there are two events, right? Karyokinesis and cytokinesis. In karyokinesis, we have prophase, metaphase, anaphase and telophase, in which the genetic material is divided among the daughter cell, which is followed by cytokinesis, in which the cytoplasmic content is getting separated among the daughter cell. So, these are the two major events which happens in the mitosis. The second question asked was, write any one difference between plant cell and animal cell mitosis. So, the question is, we have to differentiate plant and animal cell in mitosis, how it is different from each other. So, here the major difference we can able to see in the cytokinesis, that is separation of the cytoplasm, right. So, in plant what is happening, cell plate is formed and which is laid in the center equatorial plane, and the cell is divided into two parts. That is happening in plant cell. But in animal cells, the furrow formation which is happening and that is becoming constricted and that is pinching the cells into equal halves. That is what is happening in the animal cell. So, in cytoplasmic division, we could able to see the difference which is happening in the plant cell and animal cell. Question number 3 asked was, draw and explain metaphase. Any phase can be asked in the examination. So, we are seeing only one example, that is a metaphase, how to explain and draw. Okay. In metaphase, how the chromosomes are arranged. The chromosomes are present in the center equatorial plane, right. In the middle part, we can able to see the chromosomes are laid over. This is how we are representing, right, and this is an animal cell. So, we can able to see the centrioles are also present. So, here what is happening, the two chromatids, can you able to see the two chromatids which is present in all the chromosomes? We can able to see 4 arms which is present in the chromosome, which are held by the centromere, and then centrosoles are also present. This is how metaphase is drawn. And we will see the explanation. Each chromosome consists of 2 chromatids which are held by the centromere. The chromosomes arranged in the middle or in the equatorial plane. Okay, this is the explanation as per the textbook for metaphase. The next question is, write the significance of mitosis. Here, based on the answer and the marks, you have to give the answer. If it is a 5-mark question, you have to elaborate it. If it is a 2-mark question, you can mention any 2 important points. Okay, it is not randomly you can write something, correctly you mention it. So, the answer is mitosis is a equatorial division. So, the 2 daughter cells are identical to each other with that of the parent cell. That is the first significance. Second is, it helps in wound healing. Replacement of cells lasts during wear and tear. So, we can keep growing, right? We can have some kind of wound healing process is also possible. It is responsible for the growth of the organism by adding new cell to the organism. The fourth significance is, it is a method of asexual reproduction in unicellular organism like amoeba. So, we can able to get the 2 amoebas which is formed at the end of mitosis. It is a form of reproduction in the lower organism. Now, we will see today's learning objective. Today, we are going to study meiosis, stages of meiosis and the significance of meiosis. First, we will start with what is meiosis. Meiosis is present in the sexually reproducing organism, where you can able to see male and female. We can able to see in birds, you can see in papaya plant or in pumpkin plant, right. So, male and female organisms are separate. In that, the meiosis is one of the important kind of cell division which is happening. In animal, meiosis is taking place in only the reproductive organ. You can understand like it is a testis and ovary which is present in male and female organism in animals, right? When you are considering the plant, it is known as anther and ovary. In anther, we can able to see the pollen grains are started forming, and in the ovary, the ovules are getting produced. So, these are the organs in which meiosis occurs. We cannot able to see meiosis occurs in all the other organ. The stages of meiosis. Broadly, meiosis is completed in two phases. What are they? Phase 1 and second one is phase 2. So, two phases are happening in meiosis. In phase 1, the two cells with half the number of chromosomes in each is formed at the end of phase 1. That means what? The chromosome number, how much ever is present in the parent cell is getting reduced. It is becoming half in the daughter cells. And as usual, we are getting two daughter cells at the end of phase 1, but the chromosome number is not maintained. It is getting reduced to half. Hence, it is known as reduction division. Phase 1 is known as reduction division. Then what happens to phase 2? Phase 2 is as similar to as what is happening in mitosis. So, we cannot able to see the half the number of chromosome, but here the half the number of chromosome as that of the parent cell before, whatever it is getting, it is maintained, and at the end we are getting the cells which are having half the number of the chromosome of that of the parent cell, exactly like mitosis. What are all the various stages are there? Similar to that, it is all happening. So, at the end of phase 1 and phase 2, together it is known as meiosis, right? So, meiosis is producing 4 cells at the end, and these 4 cells are having half the number of chromosomes as that of the parent cell. So, what are the sequence of events which is happening in meiosis 1? We will see. We know about the chromatin fibers, right? The DNA which is present inside the nucleus in the form of chromatin fiber, it is getting organized, right? So, these chromatin fibers are again here also condensed to form chromosome, as we can able to see in prophase, right? So, we can able to see the chromosomes are forming. Now, what does these chromosomes are doing? They are arranged in matching. If we are having 2 sets of chromosomes, 2n number of chromosomes we have studied, right? So, if 2n number of chromosomes are present, we can able to see a chromosome which will have the same homologue, similar chromosome we can able to see. One we are getting it from our mother, the other one we are getting it from our father. These 2 chromosomes will have the same kind of architecture, and these chromosomes are known as homologous pair of chromosome. They are having matching pair of chromosome. These chromosomes are having the genes, right? The chromosomes are having genes, that is what we can able to see expressed as the character in an organism. So, in these what is happening? The genes are present, but it is not necessarily the same character it has to express. Say for an example, eye color in Drosophila, we can able to see different eye colors, right? Red color Drosophila we can able to see. Similar to that, we can able to see in a human some of the Mendelian character like interdigital hair or widow's peak, all those character, right? Whichever character which is dominant, we can able to see in the offsprings. That is 2 different alleles which is present for a particular character. We can able to see the color, right? Mother will be fair or father will be fair. We can able to see the intermediate color we can able to see in the offsprings. So, the homologous chromosomes are having the gene, but it not necessarily the same allele has to present. We can see in some flowers also, we can see in snapdragon, you can see pink color flower, white color flower, right? So, it is the character of the parent. Then what if the pollen and the ovule, it is going to produce the offspring, then we can able to see the mixture of combination which is coming, right? So these are the character which is present in a particular chromosome, and the chromosome which is of the same nature which is coming together in meiosis 1, that is known as homologous pairing that is happening. Then what happens at the end? Some of the chromosomal parts are getting exchanged. We are not exactly looking like our mother, we are not exactly looking like our father, right? We are showing the intermittent character, some of the character like mother and father. Why? Because the meiosis 1 happened, and then we are getting a mixture of character of father and mother which is present in our body. Then what happens in meiosis 1? Now these chromosomes are having 4 chromatid, and the nuclear membrane is disappearing now. Then the homologous chromosomes which paired before, right, in the beginning, now it begin to separate from each other, and they are going away from each other, reaching the separate pole. Now the cytoplasm also divides, and the cells are becoming 2 in number, and they are having half the number of chromosome what the original parent cell has. So the first few events are totally distinct, the homologous chromosomes are coming together, later everything is same. So now the each chromosomes are made up of 2 chromatid at the end of phase 1. Now in meiosis 2 or phase 2, what is happening? Meiosis 2 begins exactly, and how it has to do in mitosis, same thing is happening. At the end of meiosis 2, we will get 4 cells, right? Each cell now we are getting at the end of the phase 1. Now each cell will undergo division, right? So at the end we will get 4 cells, and each of the half number of chromosomes as that of the parent is still maintained here. Okay, now we will see a pictorial representation. The chromosomes are present, right, in at the beginning of the meiosis. We can able to see 2n number of chromosome which is present. These chromosomes are finding its homologous pair. Here for the diagrammatic representation, they are putting it in similar structure. Can you able to see the long chromosome and short, and it is having only one arm, right? So these chromosomes are finding its pair, and because of this pairing, what is happening? We can able to see the chromosomes are exchanging some of its gene, and now we can able to see at the end the 2n number of chromosomes are getting separated into n number of chromosome at the end of phase 1. Okay, with that the phase 1 completes. We can able to get 2 cells, but it is having, here it is 2n number was present, n is the total number of chromosome. Okay, 2n means 2 into n number of chromosome, and here it is equally dividing. So we are getting n number of chromosome at the end of meiosis 1 or stage 1 or phase 1. And in the second division, what is happening? Each cell has to divide. Can you able to see this cell and this cell, both the cells are dividing. This cell is giving rise to 2 cells. Similarly, this cell is also giving rise to 2 cells. The same n number of chromosome is still maintained here. Can you able to see the n number of chromosomes are still present? It is like meiosis, right? So the centromere will be pulled apart, and then we can able to see the chromosomes are having now only 2 arms which is present. Here we can able to see 4 arms, but here you can able to see only 2 arms which is present. This is at the end of the second division or phase 1 or phase 2 or stage 2 or meiosis 2, all are similar. So in meiosis, what is happening? It leads to formation of some of the gametes or the sex cells, and which is having only n number of chromosome, 2n which was present in the parent cell. Now it is getting reduced to n number of cells. Hope you understand this concept clearly. So what are the significance of meiosis? During meiosis, the number of chromosomes are halved. It is resulting in the gamete formation, right? So that is very much important. So in the offsprings, we can able to see the same number of chromosomes are retained. Otherwise, we will get double the number of chromosome as that of our parents, right? The same number of chromosome which in the species is restored when it is having meiosis, which is taking place during the cell division. The new combination of genes are possible, that we have seen, like the homologous chromosomes are coming together, right? So because of that, what's happening, the combination of genes we can able to obtain in the meiosis process. With that, we are coming to an end and we'll see some of the review questions. How meiosis 1 and meiosis 2 are different. Here we have to explain the number of chromosomes and the number of cells formed. How many chromosomes and cells formed at the end of meiosis 2? Only meiosis 2 it is asked. So we have to understand the question and we have to answer. Write the events which has happened during meiosis. So these are some of the review questions asked. Hope you understand this video properly. Stay connected with us and get information about other subjects as well, and see the video if you have any more clarification is needed. With that, I am coming to an end. Thank you so much and Om Shri Sai Ram.